Compared to the relatively simple feedback mechanism for control of water reabsorption through antidiuretic hormone, the regulation of sodium transport across the tubule is a bit more complicated. It's complicated because the regulation of sodium transport is tied into many different physiological functions, including blood pressure and regulation of overall salt and water balance. First, there's some complicated anatomy that we need to lay out. Specifically, we need to understand something called the Juxtaglomerular Apparatus. This complicated word simply means adjacent to the glomerulus. Here's the basic glomerulus-Bowman's capsule association, with the glomerulus fed by the afferent renal arteriole and drained by the efferent renal arteriole. The proximal convoluted tubule snakes off from the Bowman's capsule and eventually feeds into the distal convoluted tubule. Now, here's the complicated anatomical part. The distal convoluted tubule actually snakes its way around so that it loops back up and nestles itself at the base of the glomerulus. I've shown it here in cross-section. There's a section of the tubule epithelium where the cells are slightly thickened. This is known as the macula densa. Nestled within the space between the distal convoluted tubule and the glomerulus is a collection of cells known as the granular cells. This entire complicated association between the glomerulus, distal convoluted tubule, and associated cells is the juxtaglomerular apparatus. We'll just call it the JGA for short. The JGA is involved in the regulation of sodium transport via aldosterone. There's more that we need to understand, and for that we have to take a closer look at the JGA. We'll start with the Bowman's capsule and glomerulus, fed by the afferent renal arteriole and drained by the efferent renal arteriole. The granular cells are nestled at the base of the glomerulus. I'm depicting these as a mass of cells, but these are actually cells derived from the endothelium of the arterioles and glomerulus. That fact will be important in a little bit. The remaining part of the JGA is the distal convoluted tubule, and here there's a specialized area of the tubule which consists of thickened cells that are adjacent to the mass of granular cells. This is the macula densa. So now we have a better picture of the juxtaglomerular apparatus. It consists of the capsule and glomerulus, the granular cells, and the macula densa of the distal convoluted tubule. This complicated apparatus is involved in the regulation of glomerular filtration and many other things, and we'll have occasion to talk about those in a little bit. But for now, I want to focus on one of the functions that ultimately influences sodium transport in the proximal convoluted tubule. The cells of the macula densa monitor the flow rates and composition of the filtrate that's passing through the distal convoluted tubule. If filtrate sodium concentration or the filtration rate declines, the cells of the macula densa respond by secreting adenosine, which acts as a so-called paracrine hormone to the granular cells. A paracrine hormone is like an endocrine hormone, except that the chemical signal is local in extent, in this instance confined to within the JGA. In response to the paracrine signal from the macula densa, the granular cells release a protein hormone into the blood known as renin. Renin secretion is the first step of a complicated chain of events that ends up with the secretion of aldosterone from the adrenal cortex and ultimately the stimulation of sodium reabsorption from the tubule. Here's where things begin to get a little bit complicated. Let's start with our simplified tubule. We're just going to pay attention to sodium recovery here. We're going to mark the granular cells with this little blue blob here. Let's also slot in the adrenal cortex here because that's the source of the aldosterone. Renin is important because of its interaction with another peptide that is secreted into the blood by the liver. This peptide is known as angiotensinogen. It's always present in the blood plasma, but by itself it doesn't really do anything. What it does do is serve as a precursor for what's to come later. The whole chain of events starts with the secretion of renin by the granular cells. Renin acts as an enzyme on angiotensinogen, cleaving it to produce a smaller peptide called angiotensin-1. What's also important is an enzyme that's embedded in the cells of the vascular endothelium known as Angiotensin-Converting Enzyme, or ACE. In mammals, the lungs are the principal site for angiotensin-converting enzyme, but that fact is less important than the fact that this is an endothelial-bound enzyme. Angiotensin-converting enzyme converts, as the name implies, angiotensin-1 into the principal active peptide known as angiotensin-2. Angiotensin-2 acts on the adrenal cortex to stimulate the release of aldosterone into the blood, which acts on the tubule to increase sodium transport across its epithelium. This whole complicated arrangement, and it gets more complicated still, is known as the renin-angiotensin system. Let's put the renin-angiotensin system into the broader context of regulation of water balance. We start with the crucial axis of blood pressure, blood volume, the glomerular filtration rate, and the granular cells. We include the renal tubule in this too, of course. If there's a reduction of blood pressure or blood volume, this will produce a reduction in glomerular filtration rate. We're showing these reductions with blue arrows. Keep in mind also that blood pressure and blood volume are coupled to one another, although blood pressure can be controlled independently of blood volume through mechanisms like vasoconstriction. A reduction of glomerular filtration rate is one of the things that stimulates the granular cells to secrete renin. And through the complicated cascade of events that we've just outlined, to produce angiotensin-2 in the blood. The presence of angiotensin-2 in the blood has several consequences, one of which is its action on the adrenal cortex to stimulate aldosterone secretion, as we've already outlined. This increase of aldosterone acts on the tubule to stimulate the recovery of sodium from the filtrate. Angiotensin-2 also acts directly on the blood vessels, constricting them. That's the meaning of the word angiotensin, after all. This has the effect of increasing blood pressure independently of the kidneys, which now boosts the glomerular filtration rate. Additionally, angiotensin-2 acts on the pituitary to stimulate the secretion of antidiuretic hormone. This too acts on the tubule, opening up aquaporins in the tubule membrane and increasing the tubule's water recovery. Because water recovery depends upon sodium recovery, both the stimulation of sodium recovery by aldosterone and the increased porosity of the tubule membrane to water produces an increased rate of water recovery from the filtrate. Moving water from the filtrate into the blood increases the blood volume, which also increases the glomerular filtration rate, bringing it back to normal. So we see that there is a negative feedback loop. Reductions in the glomerular filtration rate ultimately produce the circumstances that restore glomerular filtration rate back to normal through increases of blood pressure and increases of blood volume. Finally, remember that the heart also participates in water and salt balance through the secretion from the atrium of atrial natriuretic factor, or ANF. Whereas the renin-angiotensin system is usually activated from reductions of blood volume, ANF secretion handles the opposite problem of there being too much blood. Here's a very quick overview of how it works. Let's establish the players, namely the atrium of the heart and the nephron tubule. If blood volume increases, this will bring about swelling of the atria. Remember that the atria are typically fairly thin-walled and elastic, so that an increase of blood volume can stretch the atrial walls considerably. High sodium concentration in the blood can also indirectly cause increases of blood volume and therefore atrial swelling. The atrium secretes ANF in response to atrial swelling, and from there it has several consequences. The first involves the glomerulus. ANF relaxes the smooth muscles of the afferent renal arteriole and constricts the efferent renal arteriole. This has the effect of increasing filtration pressure at the glomerulus, which brings about a general increase of the glomerular filtration rate. ANF also acts to inhibit sodium reabsorption across the tubule wall. This has the effect of keeping sodium ions within the tubule so that they're exported through the urine. This loss of sodium reduces the blood volume by making its osmotic potential weaker. Finally, ANF acts on the aquaporins in the tubule membrane to reduce the tubule's water permeability. Thus, water is no longer recovered from the tubule, allowing it to be exported from the body. All of these act to reduce blood volume and hence reduce atrial swelling and reduce ANF secretion, completing the feedback loop. So, the regulatory system around ANF takes care of the opposite problem from the renin-angiotensin system. The renin-angiotensin system kicks into action when blood volume is low and blood pressure is low. This increases water recovery from the tubule, returning it to the volume of the blood so that pressure can be brought back up. The ANF system, in contrast, takes care of blood volume being too high. This system increases filtration rate and simultaneously ensures that water and solutes are no longer recovered from the tubule and are exported from the body in the urine. You may have noticed a problem in our descriptions of the control of tubule function so far. The problem is that water recovery depends upon sodium recovery. That's an interesting connection, so to speak, but it does limit nephron function to the production of urine that is either voluminous and hypoosmotic to the blood, or is sparse in volume and isoosmotic to the blood. What seems to be missing here is the ability to make urine that is more concentrated than the blood, that is, making urine that is hyperosmotic to the blood. This is a problem that mammals and birds have managed to solve with their specialized kidneys, and we'll come to their solutions in a little bit. Before that, though, I want to pay attention to a problem that is inherent in the design of the fish nephron, that is, the unavoidable accumulation of sodium that follows from water recovery from the filtrate. Reducing urine volume, conserving water really, means powering reabsorption of water by the active reabsorption of sodium. The problem comes in because the adaptive need to conserve water usually is coupled to environments where the body is already under sodium stress, as in fishes that inhabit marine environments or terrestrial animals other than mammals and birds. So there's a whole separate suite of adaptive problems that arise from the way the nephron handles water recovery.